Welcome back, anatomy students. This week's lecture will be over the respiratory system. Our old friend Ibn al-Nafis is credited with first describing pulmonary circulation, the connection between the heart and the lungs, which we briefly discussed in previous lectures. Our focus this week will be on gas exchange between the incoming air and the blood. We've already learned how the heart moves the blood and a bit about the path the blood takes, but now we are going to look a little bit closer at the structures involved in oxygenating our erythrocytes. The left side of this animation shows you air moving into the lungs during inspiration, out during expiration, and the changing pressures that facilitate this process. To the right is a magnified view of the lungs showing the alveoli. These sacs are surrounded by capillary beds, and this is where gas exchange occurs. The respiratory system has about 1,500 miles of airway and between 300 and 700 million alveoli, which can provide 70 to 120 square meters of surface area for gas diffusion. As always, let's start by identifying the functions of this system. First, the main function is gas exchange of oxygen and carbon dioxide. Second, the respiratory system helps with speech. Third, it is involved with olfaction. You studied this in 201, so we won't be going over this in lab. And last is pH control. Again, we won't be going over this in lab, but you will in lecture. Some of the major structures you will need to know are the nose, the nasal cavity, the pharynx, the larynx, the trachea, bronchi, and of course the lungs. As always, let's start on a larger scale and get more detailed as we go on. We'll start with air coming in through the nose and work our way to the alveoli. The nose has two external nares that allow air to pass into the nasal cavity. Just inside the nares is the vestibule. This region has guard cells that offer protection from incoming particulates and can be a distraction when someone forgets to trim them back. Thanks, Grandpa. Behind the vestibules is the nasal cavity. This is covered with pseudostratified ciliated columnar epithelial tissue. Together, the mucus from the goblet cells and the cilia help trap dust and particulate matter, preventing it from getting into the lungs. Inside the nasal cavity are those nasal turbinates you learned about in 201, the superior, middle, and inferior nasal conchae. Do you remember which one is a separate bone? And do you remember which bone the other two are a part of? If you said the inferior nasal conchae is a separate bone, you are correct. This means the superior and middle nasal conchae are part of the ethmoid. These turbinates warm and humidify the incoming air as well as create a turbulent flow which helps in olfaction. Just inferior to those turbinates are the internal nares that lead into the pharynx. Here are a few fun facts about sneezing. The title is hyperlinked to a slow motion video of a person sneezing. You don't need to know this for the test, it's just here to help you better understand your body. Sneezing was once thought to travel about 100 miles per hour, but as of 2013 has been shown to travel only about 10 miles per hour. That being said, particulates can travel up to 24 feet and be suspended in the air for up to 10 minutes. If you're like me and tend to sneeze when you walk outside or look at a bright light, you are of the 20 to 30% of people who experience photic sneezes. Two nerves you might remember from 201 are the optic nerve and the trigeminal nerve. The optic nerve sends signals to the brain from the eyes, and the maxillary branch of the trigeminal nerve stimulates the brain to trigger a sneeze when it is stimulated by an irritant in the nose. It's thought that when a person enters a bright environment after leaving a dark area, the signal from the optic nerve crosses over to the maxillary branch and stimulates a sneeze reflex. You can think about that the next time you walk outside to catch the bus. The pharynx is the next stop on our magic school bus ride. As air is inhaled through the nose, it enters the nasal cavity, is warmed, humidified, some particulates are removed, and because of the angle leading into the pharynx, air slams into the pharyngeal tonsils. Tonsils are our first line of defense against pathogens entering our airway. Particulates and pathogens get caught in the tonsillar crypts and can elicit an immune response because of the lymphatic nodules that line the crypts. If you haven't watched the online lymphatic system lecture and completed the online activities yet, you are slowly running out of time. Next, air moves into the nasopharynx. Like the nasal cavity, the nasopharynx is lined with pseudostratified ciliated columnar epithelium to trap debris. This is also where the eustachian tubes join from the inner ear, equalizing pressure between the outside and inside environments. The next region, highlighted in pink, is called the oropharynx. It is lined with non-keratinized stratified squamous epithelial tissue to create a moist, abrasive-resistant layer that protects you from sharp pieces of chips, food, or even swords you might try to swallow. The oral cavity has palatine tonsils up here and lingual tonsils at the base of the tongue. Together, these trap foreign invaders coming in through the mouth. The uvula triggers our gag reflex, and together with the soft palate, blocks off the nasopharynx during swallowing to prevent food from going the wrong way. The epiglottis is elastic cartilage covered with a mucous membrane, and like the uvula, covers the opening into the airway called the glottis. During swallowing, this directs food into the esophagus and prevents food from entering the airway. This blue region is called the laryngopharynx. It is lined with pseudostratified and stratified squamous tissue because both air and food are passing through this region. And back here is the larynx. We are going to continue our trip through the larynx, also known as the voice box, and follow air down towards the alveoli. But first, this is one person who doesn't seem to have an issue with a gag reflex. The larynx is made of hyaline cartilage and is involved in breathing, making sounds, and helps keep food out of the airway. For reference, here is the hyoid bone. The epiglottis, seen from this anterior view, covers the glottis during swallowing. The thyroid cartilage is often seen protruding in males and is commonly called the Adam's apple. It increases in size in the presence of testosterone. Below the thyroid cartilage is the top of the trachea called the cricoid cartilage. This is the only complete cartilaginous ring and acts as an attachment site for ligaments, cartilage, and muscles and facilitates an opening and closing the airway during speech. Below that are the tracheal cartilages. These are C-shaped rings connected at the ends by the trachealis muscles. From the posterior view, you will need to be able to identify a few other cartilages. Again, here is the epiglottis, and here is that cricoid cartilage. Attached to the cricoid cartilage are the arytenoid cartilages. They are responsible for abducting and adducting the vocal cords. On top of the arytenoid cartilages are two horn-shaped cartilages from which they get their name. They are called the corniculate cartilages. From a mid-sagittal view, we can see all of these structures, but also the cuneiform cartilage. These provide structural support to the vocal cords. So let's look a bit closer at the vocal cords now. In this cross-section, we can see the thyroid cartilage here with arrows indicating the anterior side. The vocal cords are attached to the thyroid cartilage anteriorly and posteriorly to the arytenoid cartilages. As the arytenoids move, they abduct and adduct the vocal cords, which stretches and relaxes the cords, changing their pitch. During adduction, or the left image, the pitch will be higher, and during abduction, or the right image, the pitch will be lower. The vocal cords are covered in stratified squamous tissue to withstand the vibration and contact with each other. Men tend to have thicker, longer, looser cords, which also thicken during puberty in the presence of testosterone. Women tend to have thinner, shorter, tighter cords, hence higher voices. This is an image from an endoscope showing many of the structures we have talked about so far. The title is hyperlinked to a video that shows an endoscope of a woman's throat. As she sings and changes the pitch of her voice, you can see her vocal cords vibrate and change in shape. You should be pretty familiar with this slide, but here you can see the C-shaped hyaline tracheal cartilage. The esophagus, drawn in here, lies posteriorly to the trachea. And here is the trachealis muscle. During a cough, this muscle contracts, slightly constricting the tracheal cartilages, which helps expel air more forcefully. You can also see the pseudostratified epithelial tissue lining the trachea. Last, here's that same image under a microscope. Pause the video here and see if you can identify the trachea, the hyaline cartilage, the esophagus, the trachealis muscle, and pseudostratified epithelium. Let's go a bit deeper into the body and follow the pathway that air takes. Here's the trachea. This last cartilage of the trachea, called the carina, kind of looks like a pair of tighty-whities. The carina has tiny hair cells inside and is a last resort cough reflex if something makes it down this far. The trachea splits into two main bronchi. The main bronchi split into lobar bronchi, named for the lobes of the lungs, which then split into the segmental bronchi. To clarify, the singular is bronchus and the plural is bronchi. The bronchial tree refers to the extensive branching of your bronchi, also called branching generations. In adult humans, there are about 23 branches. We've only covered 3 so far. But don't worry, you don't need to know all 23 branches. Here is a plasticized bronchial tree and trachea from a Body Worlds exhibit. And this is apparently an X-ray of someone that had inhaled a pea and it lodged in the person's bronchus. Usually, when items are inhaled into the bronchi, they tend to go to the right side because the right main bronchus tilts down at a greater angle as compared to the left main bronchus, which comes off the carina at more of a right angle. Here's a quick animation of a set of lungs being inflated. There are so many air pockets in lungs that they will actually float in the water, kind of like a pumice stone. The lungs are divided into lobes, and the right lung is slightly larger than the left because of the heart's positioning. At rest, an adult breathes between 12 and 18 times a minute, or 17,000 to 26,000 times in a 24-hour period. And as we'll soon see, about 30% of the air never reaches the alveoli and stays in the airways. This is called dead air. It's useless for oxygen diffusion because it doesn't contact simple squamous epithelial tissue. One other piece of information you should be aware of is exhaled air contains water vapor from our bodies. Each day we lose about half a liter of water just by exhaling, and the higher you go in elevation, or if you live in drier climates, you will lose even more water. Here you can see the right superior lobe, the right middle lobe, the right inferior lobe, the left superior lobe, the left inferior lobe, and down here is the cardiac notch. I bet you can't guess what lies in there. This image is here to show you what the lobes of the lungs look like on a cadaver. You can distinctly see each lobe and the cardiac notch just below the left main bronchus. Okay, so similar to what we saw with the heart, the lungs are surrounded by pulmonary pleurae. The first layer is the outer parietal pleura that adheres to the chest cavity wall. The next layer is the visceral pleura, the inner serous membrane that adheres to the surface of the lungs. Between the parietal and visceral pleura is the pleural cavity, the fluid-filled space. It decreases friction during inhalation and exhalation, compartmentalizes the lung, reducing the spread of infection, and as we'll soon see, helps with inflation of the lungs. This next slide makes for great test questions. You'll need to know the order of structures during an inhalation and exhalation for your tests, so practice the flow of air forwards and backwards. As air comes in the nose or mouth and down into the trachea, it passes into the bronchi. These structures are reinforced by cartilage to help keep them open. The main bronchi divide into lobar bronchi, which divide into segmental bronchi. The segmental bronchi, also called the tertiary bronchi, divide into bronchioles. Bronchioles are much smaller and lack the cartilaginous reinforcement. Instead, they have a simple columnar epithelial lining surrounded by a layer of smooth muscle and are held open by surrounding elastic fibers. When the smooth muscle contracts, pressure increases to force out air. These bronchioles end in terminal bronchioles, which signify the end of the conducting zone, or the part of the respiratory system that moves air. This correlates to branches 1 through 16. Remember, don't worry, you don't need to know the numbered branches. The terminal bronchioles then divide into respiratory bronchioles and signify the start of the respiratory zone. This is the region, branches 16 through 23, where oxygen and carbon dioxide diffusion occur. These structures have simple squamous epithelial tissue lining the inside and have some alveoli connected to their walls. The respiratory bronchioles branch into alveolar ducts, which lead to alveolar sacs, or the clusters of alveoli. They look similar to a bunch of grapes. And finally, an alveolus is the site of 90% of gas exchange. Alveolus is singular, and alveoli is plural. In this 20-second animation, you can follow an erythrocyte on its path through the systemic and pulmonary circuits. There is also an ECG present, so you remember to build on your previous knowledge. As the erythrocytes make their way to the capillaries in the lungs, oxygen from the air we breathe diffuses into the blood, binding the hemoglobin molecules. At the same time, carbon dioxide diffuses from the blood into the alveoli so it can be expelled during an exhalation. The oxygenated blood is then transported back to the capillaries of the body. Once in the capillaries, oxygen diffuses into the surrounding tissues to nourish the cells, and carbon dioxide, a result of cellular respiration, diffuses into the blood, and the process repeats itself. While you study this process, connect the electrical system stimulating the heart, the mechanical processes of the heart pumping the blood, and all the vessels it is transported through. Here's a histological view of the lungs similar to what you will see in lab. Here is an alveolar sac, and over here is a single alveolus. Down here is a bronchiole. You can see the smooth muscle layer superficially and the inner pseudostratified layer creating the lumen. And over to the right is a blood vessel containing erythrocytes. Can you figure out if it is an artery or a vein? I can't express how important it is to practice your histology. Understanding what tissue is present will help you identify structures and locations in the respiratory system and benefit you on your tests. Be sure to look at multiple slides in lab and practice in open labs. In this next section, we will study the mechanics of breathing, different lung volumes, and a few pathologies. To clarify, breathing is the process of moving air into and out of the lungs through inhalation and exhalation, while respiration refers to the process of oxygen and carbon dioxide moving between the lungs, blood, and tissues. Let's look at the mechanics of breathing first and what happens along the way to help us move air in and out of the lungs. During an inspiration, the diaphragm contracts due to stimulation from the phrenic nerve. As it contracts, it flattens out, increasing the volume of the thoracic cavity. The parietal and visceral pleura stick together because of the serous fluid in the pleural cavity, and that expands the lungs. Volume and pressure are inversely proportional, so as the volume increases, pressure inside the lungs decreases slightly below the external environment by about 3 millimeters of mercury. As the pressure drops below the external environment, air moves into the lungs. During an expiration or exhalation, the opposite happens. The diaphragm relaxes and returns to its domed shape, decreasing the volume of the thoracic cavity, increasing the pressure slightly above the external environment, causing air to move out of the lungs. In lab this week, you will be doing a respirometry activity and calculating different lung volumes. Before you dive into that, let's look at what these volumes look like on a graph and define each term. We'll look at the full graph after we go through each volume. The first volume is called tidal volume. This is the normal volume of air that as you sit there watching this video, you are inhaling and exhaling. In this graph, you can see that the tidal volume is approximately 500 milliliters of air. The second volume you'll need to know is called inspiratory reserve volume. It is abbreviated as IRV and is the maximum amount of air you can inhale after a normal inhalation. It is important to distinguish that last part, after a normal inhalation. Remember, this is a different volume we are measuring, so we want to know how much air you are able to inhale on top of what you normally inhale. This graph shows a person with an IRV of approximately 3,000 milliliters. The third volume is the expiratory reserve volume. This is the maximum amount of air one can exhale after a normal exhalation. So again, after a normal exhalation is key. It is a measurement of what you are capable of forcefully exhaling out of your lungs. In this example, about 1,000 milliliters of air. The vital capacity is the total volume of air that can be forcefully expelled after a maximum inhalation and is calculated using the equation IRV plus ERV plus TV equals vital capacity. You may have noticed that this was a capacity and not a volume. Capacities are combinations of volumes. As you can see in this graph, the vital capacity includes inspiratory and expiratory reserve volumes and the tidal volume for a total of about 5,000 milliliters. The last volume to know is the residual volume. This is the amount of air that you just cannot get out of your lungs, no matter how hard you try. You can see in this example, it is just over 1,000 milliliters. This air helps prevent the lungs from collapsing. Now to clarify, this is not the same as when you get the wind knocked out of you. Getting the wind knocked out of you is actually spasms in the diaphragm because of some force or impact to the abdomen or solar plexus. This impact paralyzes the diaphragm for a couple minutes, making it hard to breathe. To review, here is the full graph. You can see that the graph is made of individual volumes that can be added together and referred to as capacities. Make sure you practice these volumes for your test next week, and as you go through the activity, be sure to use the terms while the subject is using the respirometer. Some pathologies you will learn are divided into obstructive and restrictive. With obstructive diseases, exhalation is mainly affected. This can make it more challenging to exhale, or it may slow exhalation down. Some examples are asthma, COPD, or cystic fibrosis. It can feel like you're breathing through a really thin straw. With restrictive diseases, the maximum volume is affected and the lungs can't expand as much. This can be a result of anthracosis, obesity, scoliosis, or muscular dystrophy. You can feel like there's an elephant sitting on your chest when you try to breathe. Emphysema is a type of COPD, or chronic obstructive pulmonary disease. Smoking is the #1 cause of emphysema, and COPD is the 4th leading cause of death in the United States. With this disease, air sacs are destroyed, which reduces the amount of simple squamous tissue in the lungs, reducing the body's ability to exchange gases, essentially starving the body of oxygen and toxifying the body with carbon dioxide. Emphysema is not curable, but it is definitely preventable. In lab, you will be looking at a slide of a lung with emphysema. On the left, you can see lots of surface area because of the abundant simple squamous tissue making the alveoli. On the right, you can see there is much less tissue, less alveoli, and therefore less surface area for gas exchange. This image shows you the difference between a non-smoker's lung and a smoker's lung. Can you tell the difference? And one last video demonstrating the inflation of a smoker's and a non-smoker's lungs. Again, can you tell the difference? Some of you may have asthma and had to deal with the spasms that occur in your bronchioles. It can be scary depending on the severity of the attacks. The smooth muscle around the bronchioles contracts and constricts your airways. The airways can also become inflamed, narrower, and produce mucus, making breathing difficult. The last disease we will look at is anthracosis. This disease is the milder version or initial stage to pneumoconiosis, or black lung disease. Coal dust that enters the lungs can't be removed or destroyed. And as a side note, if the cilia of the pseudostratified tissue is destroyed, either by smoking or other air pollution, then more dust is able to enter the lungs. Macrophages engulf the particles and eventually leads to inflammation and fibrosis or scarring, thus thickening the alveolar walls. In these images, you can see the black aggregations of macrophages containing the carbon particles. To be clear, you will see black deposits in the slides of emphysema, but what should be apparent and what you will want to use to tell the difference between the two is the lack of tissue in emphysema and the thickening of the tissue in anthracosis. If you're interested in learning more about coal mining, its influence on politics, the environment, and human health, check out these 2 documentaries. So that's it for this lecture. When studying the slides in lab, be sure to look at different slides of the same structures so you understand their variability. Your lab TA will go over the instructions for your respirometry activity in lab. As you work on this activity, use the vocabulary with your lab mates and apply the concepts you learned in this lecture to the activity. As always, keep up with all the drawings, reviews, and online lectures, and I'll see you next week for the urinary system.